Thanks so much to both Jim and Mark for getting the course started. We've covered a lot of territory in a short amount of time. This is terrific. So we do have some questions that have come through. I'm going to go ahead and start with Jim. The first question, you mentioned that the study population may be narrowly or more broadly defined. Other than enrichment, can you say more about considerations for each of these?
Sure, Kim. Thanks a lot for passing along that question. Yeah, so the study population can be narrowly defined at times or more broadly defined at others. And I think I gave some examples of this, but I'm happy to embellish on it a little bit. So I'm going to think about it from the context of a typical drug development program here, but the principles could apply to other clinical research. So early in drug development, when a drug is first being studied, for example, for a particular use, one is probably doing trials that involve a fairly small number of patients to test a drug, perhaps to explore certain endpoints, to get proof of concept, figure out whether this drug candidate is worth developing further for a particular use. And so really one is trying to identify a signal and really find out whether or not, there's a signal here to carry that drug further in development. And so when we think about how to best define a signal, you can either have a drug with a very large effect, or what's more common is to have a drug that might have a smaller effect, in which case you really want to be able to distinguish it from the noise. And so reducing noise or reducing variability becomes particularly important to try to help identify a signal. And so in early development, one approach to reducing variability is to more narrowly define a patient population such that there is more homogeneity in the population that you're exploring the drug in. And there's obviously limitations to that, and that is you're answering questions about a fairly narrow population that, as the drug is then carried further in development, you might realize that it does not work as well as you hoped once you broaden the population out. But that's one example. Another example could be from a safety perspective. Early in development, when there's not as much known about a particular new drug, you might have very real and legitimate concerns about exploring that drug in patients with certain comorbidities or other complicating features, because you're trying to get not only to protect the patients before you have more safety data accrued, but also it might be more difficult to discern what that drug's safety profile is, on a background of other comorbidities, other concomitant medications and whatnot. But as I mentioned, as a drug goes further along in development, one is not going to want to continue to restrict the population to these fairly narrow groups, but rather really broaden out the population, so that by the time one is putting together an evidence package from both an effectiveness and a safety perspective, the drug has really been studied in the patients for whom you would expect to take the drug if that drug were to be approved. And I would think that the same principles would apply elsewhere in clinical research, that you really want the results from the clinical investigation to be generalizable to whatever the clinical research question that you've asked, and not only applicable to, for example, a very narrow subset of individuals. So hopefully that helps answer the question. Thank you.
Thanks, Jim. And I have one follow-up question that is related. Would the use of enrichment in selecting the study population lead to a, quote, tighter label?
Thanks, Kim. That's a good question too, and you're right, very very related. So if one were to progress in drug development and use various enrichment strategies, and you generated evidence from clinical investigations in a particularly enriched population, the question is really what scientific conclusions can then be drawn from that, which is really how, if you're seeking drug approval, how labeling um, would be written, because the labeling should really reflect the use of the drug as it's been demonstrated to be safe and effective. So one example would be, and there's really, I should say, there's no just rules of the road here. Sometimes, and by that I mean that it's very case specific. So, but I'll just give you a couple examples that will hopefully illustrate the concept. So let's say one is using a predictive enrichment strategy where you have a drug, as I included in my challenge question, that perhaps has a mechanism that is targeted to a particular, gene variant of a disease. Well, one would then expect for that, the presence or absence of a particular variant to be predictive of whether that drug would work. And if that is in fact the case, then the labeling would very appropriately be limited to the patients for whom you expect the drug to work. And this can be a little bit difficult because it's not always obvious. And sometimes perhaps data are gathered on a biomarker positive, for example, population, and based on very good and solid hypotheses, and you don't have as much data in the biomarker negative population, and perhaps the drug is actually still effective, just less so in biomarker negative patients. And so really the extent to which one actually has data on those populations to see if there could be an interaction between, for example, biomarker positivity and the treatment effect, would all play into decisions about how the label might be written. With respect to prognostic enrichment, it's also not, there's no one size fits all answer. Sometimes it's very reasonable to enrich a trial for a high risk population because events will happen faster, but there's no scientific reason perhaps to think that the drug would not be effective in patients who are a bit lower risk, maybe moderate risk or whatnot. That maybe the effect size or the absolute risk reduction, for example, would be smaller in the low risk patient. And those are really scientific considerations that one would have to think through. I can think of examples, where prognostic enrichment has led to a labeled indication that is exactly the population that was studied in a trial, and other situations where it was believed that the indication could be broader and not limited to the quote unquote high risk patients, but other sections of the labeling would describe the enrichment strategy, so that providers would have that information should they choose to take a look at it. So hopefully that helps, and thanks again.
Great, thanks, Jim. And Mark, we have a couple of questions for you here as well. First, can you say something about what makes a good endpoint in a clinical trial?
Oh, thank you for that question, Kim. Yeah, no, that's a very good question. Dr. Jim Smith addressed some of that in his talk, that first and foremost, it should address the clinical question. The endpoint should be chosen to get at what question you're asking for. But there are definitely more considerations that come into play. It should be what we would call sensitive to the drug. So, for example, or sensitive to the perceived effect of the drug. For example, if you're maybe studying an antidepressant and you don't expect to see an effect by week 2, but maybe the mechanism of action or past experience would lead you to believe, excuse me, you would see effect maybe at week 12, then obviously week 12 is the more appropriate time point for the endpoint. So you're trying to look for something that would be sensitive to the hypothesized drug effect. Now, it still has to be clinically relevant. If reversing that example, if the effect only had an effect on depression, if the drug only had an effect on depression at 1 week and no effect beyond 1 week, well, you might be able to show an effect, but it probably wouldn't be very clinically relevant. So again, the first criteria has to be clinically relevant. But moving on from that, being sensitive to the hypothesized drug effect is important, and then the bias and variability come into play. As I said, you're always trying to minimize bias and minimize variability. Here, variability would mean, like, if for some reason you were theoretically able to take the measurement twice, would you get nearly the same result, or is it going to be very variable? Some endpoints are just naturally going to be not very variable. Maybe a heart attack is a very hard endpoint, and how you measure it, if you were to go back and measure it, take the labs and EKGs, you would get the same answer. Other endpoints may be more variable. Endpoints based on maybe a patient-reported endpoint might be more variable. If the patient took the same answered the same questions again, they might come out a little differently. But again, you have to foremost stick with the clinical relevance. So variability is important, but you don't want to answer a question that's no longer relevant. Just one kind of closing thoughts on this is FDA does have a series of guidances and draft guidances on patient-focused drug development. They're mainly focused on the issues of these, maybe something like a patient-reported outcome, although there's other examples in those as well. But they do go into a lot of what makes a good endpoint, both from a clinical relevance point of view and including bias and variability. So I hope that kind of gets at some of at least my thoughts on what makes a good clinical endpoint.
Great, thanks, Mark. And one additional question for you. If a study is not statistically significant, what can we conclude? Can we say that the drug has no effect?
Okay, another good question. I mean, this is a question that goes beyond even clinical trials, but I think it's an important thing to consider. So the concept of statistical significance is a tricky concept, and that's unfortunate. That's unfortunate for you non-statisticians, and it's unfortunate for a statistician. It means, as I tried to explain in the talk, that you've observed something that's very unlikely if the drug had no effect. So therefore, you conclude it probably did have an effect. So when you have a statistically significant effect, you can generally conclude the drug has an effect. I mean, of course, you have to consider all other issues of biases that may have resulted in this. But let's assume it was a very clean, well-conducted, simple trial, and you see a statistically significant effect. Great, you see effects, so you say the drug has an effect. Now, the converse is more tricky. If something is not statistically significant, you cannot automatically conclude that the drug doesn't have an effect. There are several things that may come into play here. Maybe the most important to consider is what I tried to get at, is statistical power. It may be that the study had very little power to detect an effect. So even if there was an effect there, the power to detect it might have been very low. The power is a number, is a probability itself. So it can go from 0 to 100%, although it probably can't be either 0 or 100. But typically we design clinical trials that have 80 or 90% power for a given effect. So if we think the effect, say, is 2, whatever that might mean, we have 80% or 90% chance of detecting that. But if we run small trials or trials that are underpowered, we may not detect real effects. So the lack of statistical significance doesn't necessarily imply that there's no effect. Now, if the trial had a lot of power, that's already giving you evidence that if you didn't see it, then it probably doesn't have an effect. But maybe a more straightforward way to examine that is to look at confidence intervals, which I didn't go into in my presentation. But a confidence interval gives you the range of likely effects. And at the upper end of the likely effects is still a very small effect, then you might conclude that the drug doesn't have much of effect. So again, I hope that addresses this question. Thank you.
Yes, thanks, Mark. And I think we may have enough time for maybe one more question for each of you. Let's go back to Jim. Is placebo the same as no treatment? If not, what's the difference?
Thank you for giving me the chance to clarify this. So absolutely true that placebo and no treatment both mean that someone is not getting an active drug. The difference between a placebo-controlled trial and a no treatment controlled trial is really blinding. So in a placebo-controlled trial, as I described, and I think as folks are probably well familiar, that is going to be a blinded trial because otherwise you're giving participants a placebo and telling them, hey, I'm giving you a placebo. That might not be the effect that you're intending to have and could introduce bias into the trial. So a no treatment control trial is really when blinding is not possible and it's going to be an open label trial anyway. Then you still have a control arm that's just getting standard of care, and then your interventional arm is likely getting standard of care plus the open label intervention. So hopefully that clarifies. Thanks.
Great, thanks, Jim. And Mark, one more question for you. How can data fudging, that's a quote, be avoided, and how can you detect if it happens?
Okay, another good question. And I think during my talk, I probably gave the primary way of avoiding it, but I'll go deeper into this. That to avoid data fudging is really a problem multiplicity. I assume that's what's meant here. I assume it's not meant like making up numbers or so. I think what's meant here is that you've looked at a lot of results. Maybe you've conducted a lot of studies, and just like my example, you found one subgroup perhaps that was statistically significant, and then you write a paper on that, and you don't mention all the other things you looked at, and you just mention I have a statistically significant result. That's what I think is meant by fudging here. And as I said in the talk, the best way to avoid that is pre-specification. That you have a protocol in place, and hopefully in a statistical analysis plan if the analysis is non-trivial. And this way, if you set out ahead of time what you're most interested in, and if you find that, it's an important result, and if you don't, that might be interesting to report as well. But if you start reporting on other things that you didn't set out to look at, you have to come clean about that this is an I think Dr. Jim Smith went into this. This is an exploratory endpoint. Although the primary endpoints were not statistically significant, or maybe they were, this exploratory result might be hypothesis generating that we might want to explore in further studies. Now in practice, this is a group for clinical trials. I think it's relatively straightforward. I think there may be requirements for publicly registering your protocol ahead of time so the public knows what you're looking at. I'm not personally familiar with those requirements, but that's one way to be clear what you're looking at. I mean, you could run a lot of studies and not tell people you're running a lot of studies, and then just pick out the one that's significant. That would also be kind of an issue with fudging, even if you had protocols. I think it's an unlikely scenario for clinical trials because of they're expensive and time-consuming to run. You're not running a lot of trials and just picking out the one that looks best. But I did point out for observational studies, you might see a lot more of that. Now, this group might not be generally focused on that, but it's something to be aware of from kind of a scientific point of view. So thank you. Thanks for the question.
Thank you, Mark, and thank you to Jim also, both for your presentations and for your willingness to engage with the audience with questions. And thank you to the audience for your attention and for submitting some questions for us.